Okay, so.
Except people that just late.
They have to. Maybe you sit close.
We have the patrons from Fluxus who supported the production. They wanted to return the last day of the show.
Okay.
They are traveling in France.
Okay.
Fluxus is organizing a trip to Marseille and then
Nice.
Nice. Saturday there is the ambassador from Switzerland, which is going to do the tour. Yeah.
Do you have any news of the call for proposal that we...
Yeah, unfortunately.
We didn't get the news, no, we didn't get it. But we can apply again. Yeah. I don't know when it is though. I think it might... Yeah, I was thinking it's just like every year, but maybe it's twice a year. I can't remember. But maybe it's good if we have one time.
Yeah. Because it wasn't very...
Yeah.
It was like 2 days when we knew.
And I wonder if maybe, I think they want like the research, scientific research part.
Yeah.
Has to be like very complete. Like,
Yeah.
I think.
Yeah.
They want something
because it's science giving us money.
also a new scientific discovery.
Yeah.
Or something like that. Not only, I think the scientific part has to be.
Yeah, that has to be stronger because it's funded by science.
Yeah,
so.
because it was just me and Liz. Just put our name, but our involvement in the project was...
Yeah, we need to have some software to figure it out. Yeah.
Bonsoir à toutes et à tous. So tonight the discussion will be held in English. So welcome to Jeudis Panacée in partnership with the newspaper Midi Libre. So I'm Alexis Loisel-Montambaux. I'm exhibition assistant at MoCo and co-curator of the season Art and Science. So you probably have seen the exhibition Sense Unknown, Éprouver l'inconnu at MoCo, where we can discover the works of Jerry Holder, a new installation produced for the exhibition titled Charybdis. So Joey, you're an artist researcher. You recently started a PhD exploring the notion of open worlding, what you do in your works actually, where you're opening worlds, blurring human boundaries, and often playing with our desire of controlling technology, but also controlling human other living forms. And you play a lot with our primal instincts, such as our fears, especially for deep sea worlds, and more broadly our fear of the unknown. And so your work has been shown in numerous biennials, such as the Athens Biennial, Sydney, Ljubljana, just to name a few. And so Charybdis is your first installation produced in an institution in France. It was produced by MoCo. We had the support from Fluxus Art Projects, and you work in collaboration with Benoît Charlot. So Benoît, you're director of research at Institut d'électronique et des systèmes at CNRS and Université de Montpellier. And your research crosses several fields, including microfluidics and neurology. And so you're also a member of the scientific board of this Art and Science season at MoCo. And preparing the talk, I was remembering we were along with Pauline Faure and Anya Harrison, and discussing about Joey's work during the committee. And you seem immediately extremely enthusiastic about the work, and we could definitely see some common sensitivities between both of your research. So before going into the roots of your collaboration, could you maybe we start with you, Joey, just present your research, some of your works, and then I'll ask the same for you, Benoît. Thank you.
Thank you so much, Alexis, for inviting me for this talk. Thank you, Benoît. Yeah, it's a huge honour to be in the exhibition, Sense Unknown. There's loads of great artists in the show. If you haven't seen it yet, go and see it. Also some real heroes in there. To be in a show with H.R. Geiger, the artist that made The Alien from the Aliens film, is also a dream come true. So yeah, it's great. So yeah, I'll talk a little bit about my work, some previous works, a bit of background on my research. Yeah, probably about 15 minutes or so. So yeah, I'm an artist. Like, I make multimedia installations. I work online and in an offline environments. I prefer to kind of think of my work like as a form of worlding. So I make experiences, environments, situations. I guess I think of them as kind of like film sets rather than exhibitions. And yeah, they don't just exist in art galleries. This one on the screen was actually in a deserted swimming pool. And I want them to, yeah, be these kind of immersive experiences and be part of a kind of multi-layered narrative. As Alexis said, I've just started my PhD, which is about world building in art practice, and specifically what I'd like to call open worlding, which is where artists open up their work to some kind of outside forces. So this is not a standard or recognized term within academic literature, but is related to open world design in video games, open systems theory, and the kind of philosophical ideas around world making. And I very much, yeah, want my kind of works to be like porous and plural environments where I'm not completely in control of the outcomes of the of the work as well. So I let kind of other agents and people and collaborators to come in, with the situation. So we're at MOCO, so maybe this is also related to relational art or relational aesthetics. But yeah, so a little bit of background on me. I studied art at school, but I was very close to doing science. Maybe if I'd have chosen the science route, life would have been a bit easier, but c'est la vie. When I was at school, I was taught there was like quite a rigid dichotomy, I guess, between art and science, and I was told like I'd have to choose one. But now I very much believe that they can exist and complement with each other. So some context about my work. I like to ask big questions, I guess. My work raises philosophical questions about our universe and things yet unknown across science, technology, and human-machine-animal interactions. I particularly focus on like natural history and the other life forms that we live with. And I'm very critical about how our current situation, computation, the data sphere, I guess, is creating a certain understanding about the world, a certain world picture. So often in my works, like I collaborate with scientists and other researchers, and I'll often depict creatures, often weird alien creatures, against kind of like diagrams and scientific systems, etc. Some of the brief themes about my work. I'm interested in reality crafting through digital networks, the merging of matter and information, our increased entanglement with technology, and cybernetics and world dynamics in relation to the natural world. So I'm very interested in how these systems, like our belief systems, our cultural systems, our philosophies, our scientific systems, deeply shape how we perceive the world, how we interact with it, and how we interact with our environment. And I hope that within my work that it kind of reflects a very complex, mutating reality, one that kind of avoids simple identification and classification. I don't believe that life can be easily classified or captured, but as human beings we sort of seek to do this all the time, through science or whatever else to understand it. I'll just read a quote. So this is by Henry Betson, who says, "We need another and more wiser and perhaps a more mystical concept of animals, for the animal shall not be measured by man. In a world older and more complete than ours, they move finished and complete, gifted with the extensions of the senses we have lost or never attained, living by voices we shall never hear." I think the various systems that we produce, as I say, can never quite grasp living systems. And another theorist called Karen Barrett talks about this when she says the deeply connected way that everything is entangled with everything else means that any observation makes a cut between what is included and what is excluded from what is being considered. Nothing is inherently separate from anything else, but separations are temporarily enacted, so one can examine something long enough to gain knowledge about it. This view of knowledge provides a framework for thinking about how culture and habits of thought can make some things visible and other things easier to ignore or to never see. Sorry, I forgot to do these slides. Just skip some of these. So sorry, I'm just dropping loads of theory on you. Another theorist who I love, Federico Campagna, also talks about this in a book called Technic and Magic, where he talks about like the universe and reality being like this kind of ineffable, having this ineffable quality, this magical quality that we can't, possibly realize. So yes, in previous projects, like in the lead up to the project at MOCO, a large-scale project that I completed a couple of years ago was called Cryptid, which was about mythological creatures which may or may not exist, which are unrecognized by mainstream science. And I think what's really interesting is that we don't know. Again, like related to the exhibition Sense Unknown, there's so much that we don't know. An astonishing 86% of plants and 91% of, 86 of all plants and animals on land and 91% of those in the sea have yet to be named and catalogued. So that's an incredibly huge amount. So yeah, with this kind of majority of life that's been unmapped, unobserved and unexplored, the kind of speculation for this work was like that there's millions of cryptids out there, like millions of these mysterious creatures. I was also looking into like taxonomies as well, like how we categorize certain creatures, well, all of the creatures, and how like this kind of system is very much based on this Eurocentric worldview where humans are placed at the kind of pinnacle and other life is ranked kind of below human beings. And of course this is a kind of reflection on like the European powers at the time and how this is a kind of like system of dominance, but there are lots of ways in which, you know, things can be talked about in different stories. You know, this is just one kind of system. So for the show, I like focused on like a group of creatures, plankton, which is a huge kind of like grouping of creatures, which includes viruses, algae, baby creatures, anything that's like small enough to drift along with the currents. And I chose certain plankton and then created this huge, let me just flick this on, this huge installation which ended up looking like some weird alien communication device. And yeah, it was also, I mean, again, like looking at like other kind of ways of telling stories about these creatures. I came across this cryptid core online, which is basically like loads of like fandom pages where people talk about like supposed sightings of cryptids, and everybody's kind of adding to this kind of like shared narrative, this shared kind of like internet folklore. And then the following show was more kind of conspiratorial, I guess. So I was kind of like thinking about, I guess, like the person that was searching for these mythological creatures online. So I built this kind of computer room, which had this kind of like detective wall, and yeah, this kind of like online search for this mythological creature. And then there was also like this film in the back room which was called Metis, which was supposedly this creature that had been born from the techno-capitalist regime, and it was mainly like feeding on plastic. And you followed these kind of like internet Reddit threads through the film to kind of like, figure out this story. But then it ended up that it was all made up. So I'll skip to the current show that's on at MOCO. None of us could pronounce this word. It's the name of a Greek mythology for a creature, Charybdis. And this is one of the depictions of it. And so this was the exhibition, yeah, as I said, that's here. Charybdis was a notorious sea monster embodying the perils of the nature of the sea. She became synonymous with maritime hazards and the fear of unknown depths. And for the show, like, it was quite interesting actually, like with the interpretation of the show, because as part of the show, the curator, Alexis, you wrote a text, and also Benoît, you wrote a text for it, and they seemed quite definite about the creature that was in it. So I think like you mentioned crabs and focused in on those, and then Benoît, you mentioned that it was about the discovery of a giant squid, I think. So also this like kind of like lost in interpretation of it, I really liked. I think you even asked me like, should I correct this or something and take it out? But I think, yeah, there's no correct way of explaining it. So yeah, a mood board for the work, like I was looking at people's personal computer rooms and also like control room kind of setting. So humans, maybe human scientists in some kind of lab, like looking out into some unknown, observing some unknown realm or world. And these are just some pictures and videos from the show. And Benoît like really kindly lent me some equipment as well from the lab that you also installed at the end of the show. Not the end of the show, like during the installation, so it's like this collaboration. And also Benoît gave me some imagery of insects under a microscope, and also like told me all about your work into organoids, which I won't try and explain what organoids are because you'll do a much better job of that. But I thought it might be quite interesting to finish like on these videos, because I'm sure you're going to talk more about organoids and neurobiology that you're doing. But I think like, I mean, these creatures to me look a lot like octopuses that are in the work, and they're interesting because they have like a decentralized brain. So I guess like when we think about like human, I mean, lots of people are thinking, I mean, these are AI generated. There's lots of conversations, obviously, these days about AI and consciousness, or what is intelligence. And I mean, I often looked at other creatures to kind of, yeah, I guess like question our human perceptions of this. I mean, octopuses like don't have a centralized brain, so I think like they have like many brains, so they can sense like different things at the same time. So yeah, this kind of decentralized notion of intelligence, I think like really questions our own kind of notion of like centralized embodied intelligence. Yeah, maybe I can stop there and you can take over. Oh, these are the machines, which I can't remember what they're called. Is that an oscilloscope?
Basically, yes.
I think I managed to break it.
Okay, thank you, Joey. So let me put that all in the context. So like Alexis said, we went for a meeting for the art science exhibition and group, and they present several artists, and when they show your work, I say, wow, it's cool. Just that, oh, it's so cool, I want to be part of this. There was a lot of organic stuff, a lot of technical stuff, robotic surgery, devices and things like that. So I say, oh, wow, it's something. It really resonates in my mind, and I say, probably there's something cool to do with Joey. So that's why I propose you to be part of this exhibition. And for us, for a scientist, it's a lot of fun. The first thing is cool and fun. So I will just discuss a bit about me. So I'm Benoît Charlot, I'm a research director at CNRS, working in Montpellier. I'm working on a lot of things. I'm working in engineering, so it means that I build devices, I build stuff in collaboration with other people such as biologists, neuroscientists, biophysicists, and so on. So I'm building small things, and some of them are on this picture. I mostly work with neurons in order to better understand how neurons work, how the brain works, and to recreate in vitro this neuroscience stuff. I'm also working with blood, with cancer, and with a lot of technical things, but today I will focus mainly on what we do with neurons and with blood. So you see a lot of, on this picture, a lot of different things, but I will try to focus more on what we do on neurons. So these are neurons. Actually, I forgot, but the image has been taken by Christophe Leterrier in Marseille. And what you see here is a lot of these cells, and there is no scale, but each cell is something like 10 micrometers wide, and they span, and a lot of neurites that grow everywhere. So it's a 2D culture of neurons. So we take a rat, we kill the rat, we take the brain, we dissociate the cell, and we put them on glass. And it's too bad for the rats. That's why nowadays we are moving from what we call primary cultures to stem cells. And it's kind of huge revolution now in science that we move from using animals to using human materials. And it represents better the human biology. It's better to use human cells. So now more and more my colleagues in biology, they use stem cells, cells that comes from part of your skin, for example, that are being differentiated into neurons or whatever cell you want to study. So these are neurons, and what you see here is that you have a lot of cells, that some of them are connected together. And if you want to study the interaction, the electrical interaction in between these cells, it's quite difficult. There's a lot of things I want to say, but one thing that comes to my mind when you talk about the squids, all the science of studying the electrical interaction between the neurons, because the neurons, they communicate with ions, with electricity, it starts with a squid. The first experiments were made on the giant axon of a squid, and it led to a Nobel Prize from Hodgkin and Huxley in the 50s or 60s. And they start by using a squid that has a huge axon. Axon is the small arm that prolongates from the cell body. And they managed to understand how the ions are moving from one cell to another, and how the electrical connection can be made, and how the neurons can compute and transfer informations. So there is a point, you show squids, and the squids were very important at the beginning of electrophysiology. This is the science and how to understand how the neurons communicate all together. So this is a 2D cell culture, and now let's see in action. So that's what we do in the lab in Montpellier. Here you see there is an optical way of observing how the neuron transfer the informations. So you see the color, they are blue, and sometimes they flash, and they have an increase in fluorescence. And this is a way to target and to measure the electrical activity of the cell. So what you see here are neurons communicating all together and sending information from one cell to another. So now we are able to observe this cell in live under the microscope and to observe the communication between the neurons. And what drives me in this field, because normally I'm working with electronic devices, computer science and things like that, is that it's possible to communicate, to make a link between the electronic world, the computer world, and the organic world. And the interface is a piece of metal, an electrode that you place in contact with the cell, and you can measure the electrical activity of the cell. So you can decode the information that is transferred between two cells, between a lot of cells, and try to understand how your brain will work, actually. So it amazed me, and I say, I want to work in that field. So this is an optical way of measuring the activity of neurons, but now we use, so I will have to go back. Here, the cells are everywhere. They place and they grow where they want. And we want to order them. We want to have some more order in order to study their interaction. And what we do in the lab is that we build microfluidic circuits. It's very small devices that allows to have kind of order in this 2D cell culture. So we use this kind of device that is fabricated in Montpellier, and what you can see here is that on the left you have one type of neurons. Here on the microchannels you have an axons that grow and that connect to a second type of neurons. And with this kind of device we can recreate some neuronal junctions, such as the one that you have from your cortex to your striatum, from your brain to your spine, and from your spine to your muscles, for example. And we can study the transfer of information between your neuron and your muscle when you move your arm, for example. So that's what we do in the lab. But it is still 2D cell culture, and now my colleagues from biology and a lot of people in biology are moving from 2D cell culture that is not really organotypic, that not resemble of what we have in the brain, to organoids. So maybe some of you have heard what the word organoids. So basically, it's a mini brain. It's the beginning of a brain. What could be, for example, in a fetus. At the beginning of life, the brain is very small and will grow. And this is a mini brain. It's something like 1 millimeter in diameter. It has a 3D shape, like spheroidal shape. And it's a very good model of how the brain works. Unfortunately, it is very difficult to work directly on the brain of people for some reasons. So in order to have it under the microscope, we recreate this brain in tiny way. But this is a spherical, and when we use the electrodes that we use to study the neurons in 2D, here you can see, here you have some electrodes, the black dots are some electrodes, and when you put this ball, these tiny balls on the electrodes, it's difficult to measure the electrical activity. So in the lab, we have been working at this, and we have been developing some, how to say, spiky electrodes, tiny cantilevers that, when we put the organoid on top, we impale the organoid on these tiny cantilevers. And we call it mini brain on a bed of nails. So when I say what I do in the lab is that I torture cells. But it doesn't feel anything and it doesn't scream. So we develop this kind of things, and the goal was to actually go inside the tissue, inside the organoid, and to place the electrode very close to the cells that are electrically active. And this is what it looks like. So we made it. Leah, you remember it? We spent the day trying to make one of these. So this is these tiny cantilevers. So they raise automatically. They are very small, very sharp, and we place the mini brain on top of it, and it gets inserted inside, and then we can measure the electrical activity inside these organoids. Okay, then you tell me what it's like to, what can we do with it? And one thing that we made with some colleagues from virology, for example, is that they infect this mini brain with some virus, COVID. It can be Zika virus, some virus that goes in the brain, and we can study the impact of the infection of this virus on the brain. And especially for COVID, they spend 3 years working on this. They don't find a lot of things. COVID goes in the brain, but at not so many impacts, hopefully. But this is an example of what we can do with this mini brain impaled on microelectrodes. I'm very long. I will continue, but
yeah, totally okay.
It's okay. And I discussed about the communication between electricity, the computer world, electronics, and the organic. And this is an example of what we record. So here on the top line, you see a line and with spikes, some events. And this is how the cell communicates. They send some inputs that we call an action potentials, like 0, 1 in the digital world. They send an impulse, and this impulse goes through a synapse and will excitate or inhibit the successive cell. So this is a recording of one neuron over several seconds, and you see that it creates several spikes. So this is really the interface between the biology and the electronic world, and it's amazing, at least for me. So the second part of my of what I will present you today is the physics of blood flows. And so this is something very interesting also. Some of my colleagues are in the audience, so we can discuss that later. Is how to understand how the blood flows in our veins, in our arteries, in our arterioles, and in the network that we have in the body. And it's something that is really amazing. And an example, so this is not my work, this is the work of other scientists. And you recognize this. What is it? Does someone know what is it? Louder? It's a brain, but what kind of animal? No. Close. It's a rat brain. It's a typical shape of a rat brain. So they use a very complex microscope, and they managed to make a computer reconstruction of all the vascular network in the brain of a rat. So you can see there is a lot of tree-like structures. There is a lot of arterioles that divide into capillaries, very small capillaries, and then that are connected to venules, and then finally to veins, and that returns to the heart. And the topology, the shape of this vascular network is really amazing. And with some colleagues, we try to understand how the blood flows into this very complex network of vessels. And for this, we use models, because we are like physicists, we want the things to become very simple. So we move from this huge 3D vascular network to a model, and this is the kind of model we use. On the left, the blood comes from arterioles, goes into a capillary bed, capillary networks, and then goes back to the heart by a network of venules. And we recreate this thing in 2D, and for this we build what we called replicas of vascular networks. We made it in the laboratory here in Montpellier. And what you observe here is the imprints. So this will be used to cast transparent polymer on top and to make the replica completely transparent. And then we flow blood inside and to observe how the cells, the red blood cells and white blood cells and so on, flow into the complexity of this network. And so I will show you some example of blood flowing into this thing. And of course it's black and white, because the scientists, we don't give a shit about the colors, we're just measuring things. So unfortunately you thought that blood is red, actually it's red, but it's only filmed in black and white. So what you see here is the venule part. The blood flow from the network on the left to the right, and you can see for the moment only things flowing. But if we will go deeper, you will see the red blood cells. So this is a view of something like 20 micron vessel, and you see that there is a lot of cells that are very densely packed and that flows into the capillary. And we try to understand how the cells deform, how they interact all together, and what is the impact on the viscous parameters, for example, how they flow into these capillaries, and some other example. What happens, for example, at the bifurcations? What are the shape of these red blood cells? They are very deformable, hopefully, because the capillaries are very tiny and they can pass through vessels that are smaller than the diameter of the red blood cells. And for example, there is some disease that impacts the viscosity of these cells, and if the cells becomes less deformable, it has a huge impact on your health. This is, for example, the sickle cell disease. So we observed this, and I wanted to show you because I found this is very nice. When you see blood, it's when that you cut your skin and you see this very red and viscous liquid. But when you look at it on the microscope, you see a lot of beautiful things. And for example, the deformation of the cells that flows into the capillary here, for example. Normally, the red blood cells, they have a discocyte shape, and when they are in a very small tube, they deform like parachutes. They deform like a bullet of a gun, and they have different names of deformed cells. No. Now we go back. Of course, the video don't work. Yes. So here you can see, for example, what happens at the bifurcations. And they deform a lot, and we can make some physics, and we can study the physics of the deformation of these cells. That's it. I will conclude. So, as a conclusion, I work on a lot of things, but I don't work alone. And one big difference between artists and scientists is that most of the time we work in groups, in small groups and huge groups. And I've been working for this with a lot of different people from different labs in Montpellier. Labs in biology, in biophysics, in computer science, simulations, and so on and so on. And we have a lot of people that give us money. Of course, the CNRS and the universities. Yeah. And this is another difference. We spend a lot of money. And also, for example, this association for the Fondation pour la recherche médicale, Fondation pour la recherche sur le cerveau. So if you have the opportunity, give them some money so they can pay us. Thank you very much.
Yeah, thank you, Benoît. Maybe let's come back to the pictures of the install, and I know you, Benoît, had some questions also for Joey.
Yeah.
We still have like something like 10 minutes to discuss.
I have a question for you.
Cool.
I mean, it's really, I don't know if you ever think about this, probably it's just me that thinks about this, but like in your work, I mean, you say that it's like this major step forward that when you can grow these things outside the body from 2D to 3D cultures, and when this is actually creating a more kind of complex model of the organs, in this case the brain, for this brain organoids. Like, I mean, do you ever think about like where, I don't know, this vitality, where life begins and it ends? Like, I mean, I'm just thinking of like, you know, do you think of that thing as being alive?
Again, this question of blurring boundaries.
Yeah.
Where life starts.
Of course, there are questions of ethics. Because we are not Frankenstein. We don't recreate life. But we have some masked piece of cells packed together, and they start acting like a real brain. we can observe what we call rosettes. It's the beginning of the convolution that you will have in your brain. So this is the beginning of a brain. And people now are working at developing more and more organs on a chip. So they are developing, for example, lung on chip, pancreas, I forgot the English word for this, but guts on chip, vascularization, and we are going towards something like they call human on chip. The goal is always to study the pathology, to recreate in vitro some models of people, of life, and to study the pathology and to understand the basic science behind. But of course, things will increase and in complexity, and maybe in the future we can grow something in the lab that probably could have conscience emerging into a brain organoids. For example, we don't know exactly what is the conscience.
Yeah.
But will it be able to suffer, for example? That's really a good ethical questions. So that's why in my community, they don't like that we called it mini brain.
Yeah.
Yeah.
I mean, I really recommend everybody to go on a rabbit hole deep dive on the internet about brain organoids as well, because you come across some crazy stories about like simulation theory and all of this stuff. Like if we can, you know, engineer more complex brains in the future. Yeah, there's all kind of speculative stories. So I also went on this as soon as I heard about your research. I was like, oh my God, like it completely, blew my mind. But I mean, obviously you have to address the ethics of it. But I mean, in your kind of, can I ask your personal like stance though on it? Like where, you know, like where do you, is there a, for you, are these things in vitro? Do you think of them as being alive?
yeah. Of course, it's alive.
Yeah.
It comes from life.
Yeah.
Before it was coming from rats, mouse, small animals. Now it's human materials. When we connect 1,000 of cells on a piece of glass, there is no fault. There is no. It's only communication from cell to cell. What we see is, for example, synchronous events that looks like epilepsy, but you don't have enough cells to have something that really work, that has a conscience. But things are growing, and the organoid, one problem with the organoid is there is no vascular network for the moment inside, so the organoid, they grow, but the inside is dying. Yeah. So the core is necrotic.
And how long do they last?
Yeah, they can last it for up to 1 year in culture. Yeah. So they grow, but the inside is dying. But people now are working at developing a vascular network inside to feed the inside of these balls, so that they can have more active cells inside. And this will be improved, and in the forthcoming years, we will start having medium-sized brain.
Yeah.
On chip.
So is that also the connection with your blood systems as well? That eventually,
the final goal is to mix both worlds, the part on the vascular networks and the part on the brain, and to try to vascularize a brain organoid, a cerebral organoid. And more recently, we try to develop cerebral organoid and spine organoid, so to model the connection between your brain and your spine, because a lot of disease arise from this problem of connection between your brain and the spine, and then after the muscle. So of course there is some ethics, and sometimes when you are working on human materials and that you have signals, you say, oh, it's what is happening in my mind, but in your brain you have 100 billions of neurons all connected together. So there is a huge difference between 1 kilogram brain and a few thousands of cells.
I don't know though. I mean, I'm not a scientist, but I'm like, I don't know. Like, I find it difficult to say like where we set the limits to the, like to something becoming okay. Like it's this complex, so therefore it is life, you know? Like maybe we can also argue for more, I don't know, like small organoids or like simpler life forms to also be life, you know? I don't know. I think it's can be more expansive. But it's, yeah, where do you draw the line? It's very difficult. And your research has like completely blown my mind in terms of like thinking about intelligence and stuff. It's crazy.
But I like the giant squid also. So where
why did they choose the giant squid for this? Because of its
because the squid has a very huge axon, and it was easy to put an electrode inside.
Okay.
And the communication was very slow, probably because they live in at 4 degrees C, very deep into the ocean.
Yeah.
And it don't need very fast action potential, very fast reflexes, and so it's floating. Don't need very a lot of computation. And they really work in the giant squid axon for the first experiment on electrophysiology. Yeah.
And Benoît, you gave us some views on how you consider your sales growth or sales cultures. And could you, Joey, give us your point of view, how you consider your works, your environments that you create when you mix like living forms with new technologies that also creates their own narratives?
Yeah.
How
I mean,
much
maybe I can follow on a bit from like what we was talking about before, and like this squid. I'm actually developing a new piece of work where I'm looking at Pacific creatures that one of them is an octopus, one of them is a daphnia, one of them is a immortal jellyfish, and the other is a volcano sponge. So these Pacific creatures, like I've chosen because, the immortal jellyfish, the trick is in the name that it can't die, so it regenerates its cells and it goes back to its, like a new sort of state, its new, a polyp state. The volcano sponge, like the giant squid maybe, is like living in these kind of slow, extreme environments, so it lives for like 15,000 years. And then the octopus, obviously, like what I talked about before, has this decentralized intelligence, and then the daphnia can die essentially. It becomes dormant, and then it can be woken up after hundreds of years. So, I mean, again, like with these creatures, like they defy what we would normally consider to be living, or or what we could consider life or death to be. So, I don't know. Like, I'm just again, like interested to think about like our, how we kind of like model our own cognition, but then these other creatures kind of like throw all of that stuff out of the water, like how it's kind of processed, and that, I don't know, that's just a comment really, like on that stuff. But I guess I'm kind of like, as I say, like interested in, I think with a lot of computation and science, we're always interested in like doubling ourselves, right? And our own kind of biology, and but maybe to step outside of that sometimes as well is like really fruitful, because it's like just kind of blows your mind about what we kind of like thought of it to be, if that makes sense.
Yeah, I want to say just one thing. Always we say when we are starting to work on life science, and when you have an engineering background, there is 2 questions that you have to ask before starting anything. Is first the question of energy, how the life forms take their energy, and the second thing is evolution. How did they get to this kind of solution with all the process of the evolution? So energy and evolution are the first 2 things that you have to have in mind when you study a life form and these kind of things. And in the deep sea, there is absolutely no energy, no sun, no light, very tiny amount of food, so very low level of energy. And they have time, and can find species that hasn't evolved since millions of years. So this is really the boundaries of the life forms that you can find in the bottom of the sea. And you show, and this is a huge part of your work that deals with these incredible creatures that we can find in the deep sea. Some of them are really gigantic, and we sometimes we don't know why are they so big, because there is no food. So maybe if they live for 1,000 years, they have time to grow slowly. But the gigantisms is something really weird for biologists that you can find only at the bottom of the sea. Okay, energy evolution.
And so yeah, maybe to conclude before giving the maker to the audience, we can definitely see in both of your of your talk that there is this common aspect in your research, that in all these processes that there is this relativity of knowledge, and that we are creating taxonomies, system of organizing understanding, and there is this relativity that these frames works until the new discoveries. And some people said that we are entering in a post-truth period, especially now with AI, but also how much these new technologies are also sometimes conquering some aspect of science, and how much now we are more conscious of this subjectivity that does exist not only in the art but of course also in science, and that these new discoveries are also always the next step to a new frame of knowledge. So thank you very much, Benoît and Joey. And you probably have questions. If you want to ask in French, we can also translate.
Hello. I wanted to ask a question about the more of the techniques and the biology when you put molecular cells between them, which data, which information were transferred, because it's so small. At the same time, there are a lot of them, but for it to transfer to a point to another, which data were they like floating from where? I don't know if you understand the question. Like.
Yo, so you want a lecture on the basic of electrophysiology? So the neurons, it's a cell. So the cell, there is an outside and an inside, and there is a membrane in between. And the neurons are polarized cells. It means that the electrical voltage is different inside than outside of the cell. Okay, so the neurons, they have less minus 70 millivolts of electrical difference. So that's the first thing. And then they have this long axon, it's like an arm, that grows from the cell body and that will be connected to another neurons. And the cell will open some small holes in its membrane, and it allows ions to move from the inside to the outside, and this will create an imbalance in the electrical potential of the cells. And this signal of ions will move from the cell body down to the axon and up to the synapse that is connected to another neurons. That was the question. So there is an actual electrical signal that spread from the cell body down to the termination of the axon, which is connected to the synapse. And the synapse, there will be a transfer of neuromediator that will excitate the cell that is connected to this synapse. It's a bit complex, and I don't have any support to explain you, but it's actually movement of ions in liquids
So
that transfer into potentials and voltages.
Okay, so it doesn't it doesn't have information in it.
Yes,
Maybe she's. I don't know if this is what you're asking. What kind of information are you measuring? Like
so
in your work?
we are measuring the movement of these ions, these potentials, and when a neuron is connected to another cells, it will send a pulse. That's the spike that I show you on the trace. And the second neurons will receive this incoming electrical pulse and will integrate all the signals coming from all the neurons they are connected to them, and then will send another impulse. So the information passes from one cell to another, and the communication is made by the frequency of this spike altogether. For example, when my brain sends a signal through my motor neurons, it will send a stream of spike with a given frequency that will actuate my muscles. I move like this because my neurons, there is a lot of cells connected all together that send a signal down to my brains, to the spine, to the motor neurons, and finally to the muscles. And we can decode these informations, and we can understand what the cells are sending as signals.
Thank you.
For example, if you put some electrodes into the brain of someone, in the motor region, you can decode the movements, the intention of movements that your brain is generating to actuate your muscles.
Oh yeah, was it
So this is what Elon Musk is doing?
Yes, actually, yes. Oh. I don't know if it's a good news or bad news, but yeah, Neuralink, they try to put deep brain stimulation and deep brain recording to record the motor regions, for example.
So we wouldn't have to move to do the action. Our brain could just
it's
send
already
the signal.
exists. Yeah, we can already decode your intention of movement. And for example, drive a robotic arm. They already made these experiments on people and on monkeys.
So one of the deep dives that I did on organoids, I mean, I think we talked about this as well, so this is probably not conspiratorial, it's true. So they do these simulations, or maybe it's not a simulation, so they put the organoid into some kind of computer simulation that flaps wings of like a butterfly or something, or controls a robot or something with the...
Yeah, I know. So in Montpellier, it's not with organoid, but with slices.
Okay.
We can also use slices. It's another kind of model. And they managed to use the slices to make it play the piano. So it recognizes a note and then drives the robotic hands that plays the piano. But it's a work on the neuronal plasticity, how you are able to learn something to your neuronal network. There was some other questions.
Hi, I have a question for Joey about the AI models that were used to generate the imagery and videos. And I was wondering, what kind of model did you use? Did you train with specialized data set with choose an image, or did you use more generalized models? And also, was using open source models a consideration, or did not care?
Yeah, I didn't use anything. I didn't use any clever training sets actually myself. It was just using. First of all, I created images of Midjourney, which was just blending images of. I can't actually remember. I think there was like octopus images. Various creatures together, and then like on these kind of coral backgrounds, and then animating it in a program. So, yeah, sorry, there was nothing like fancy me doing clever training AI. And the question, like your question on like open data sets, like I guess like you mean by like image scraping the internet.
Just to be fair, because we talked about his ethical issues in his job. So I was thinking maybe questioning the. I mean, I'm also using AI, so there's no judgment here. With generating AI that is used to scrap the entire internet images, but just I was wondering if somewhere in your process you're kind of questioning these ethical issues with copyrights, and especially when it's privatized company using all these things to generate
I mean, I
money.
think it's, no, of course. I mean, I think it's like really bad that there are these huge companies that are centralizing this, obviously, and you know, and able to control these huge data sets. I mean, like, part of my process in my work for a long time, I guess, has always been like using a lot of found footage, found images and stuff, like, which, I don't know, like, I don't feel like within my own work as well, like, that I kind of own my work or something. Like, I feel like I'm just a kind of filter or something, another kind of organizing filter or collage of this stuff. So, yeah, I mean, also, like, I mean, I remember once I did this work about 10 years ago, like, talking about copyright, because I was like, I don't feel like, I feel like everybody can also use any of my work if they want to sample it or something. And I remember I put the whole of my hard drive online once, like, as a torrent file. I said, you can download it and you can do whatever you want with it. But I mean, I still do think it's problematic, though, that, like, obviously companies have this kind of huge, you know, centralized power with being able to access, like, these huge data sets, obviously. But yeah, it seems to, like, fit, like, my process, as I say, because I do kind of collage and filter lots of stuff together. And I almost feel like, I don't know, like, with the advent of the internet or something, it's like, you just, like, with Google image searches, you just feel like everything has already been made. So it's like, why make anything else? Here is my raw material or something. Like, I'm not going to produce anything novel anymore because it's like this saturated content or something. But yeah. Are you using AI training models then?
Yeah.
Nice.
And I'll be really happy to discuss that with you, because I mean, I also believed with using open source, also uncensored models that you can find, you are not using all those trained data sets to give money to some private company, but more to create, as you say, like, share art with people that can also reuse it behind in a more communitary way.
Yeah.
And I also do believe they can produce way more interesting image. Also, considering your work works with those cryptid, found footage kind of style image, and I do believe the open source model that are way less trained to produce commercial images also produce very more coherent work in, like, your field. So I would highly recommend
it,
it, especially for your usage. So
yeah, I should be less lazy for that.
Actually, it's not that different. But thank you very much for
thank you
considering this.
We might have time just for one last question.
I just wanted to ask, how did you two came to meet? Like, can I just, as an art student, go into the science building in the Montpellier University?
Yes, actually, yes. It's a good thing, because I was going to tell you, if the students of the MOCO wants to come and visit the lab, because most of the time you don't have the good idea of what is science and what science is made. So if you want, we were discussing that with Alexis, if you want, we can organize a visit of the labs, of my labs and other labs. You can go and visit the clean room and all the experimental room. Of course, we can make it.
Thank you.
Yeah. And just to say on that as well, like, I mean, I've worked with a lot of scientists in the past, and often scientists are quite open to have conversations for their ideas. It's also good for you, right? Because you get to show your work in a different context, so they get, like, ref points for
cool
this.
And fun.
So yeah, they're often open to have, like, a chat. So, you know, just reach out and say hello.
And finally, so it was a collaboration between you and I, but there was a third person, a student of mine, and I think she's in the room, and I asked her, she was an intern in my lab, and I say, oh, we'll work with Joey, and we have to make a nice electron microscope image of bugs. So go find the bugs outside. And she came back with a bug, and we put it in the electron microscope and made this really funny image that you use on your installation. And it was really, really cool to see that the image we made with this young intern was used by you. So it was great. So I thank her. Emma.
Thank you very much. Thank you to both of you.
And we can discuss afterwards if you want.
Yeah, and so on vous donne rendez-vous la semaine prochaine pour un jeudi Moco Panacée dédié à l'artiste Damien Fragnon, donc sur une invitation des étudiants et étudiantes du Moco SBA. Merci à vous, bonne soirée.